morning, everyone, and welcome to my presentation. Today, I will be talking about the clinical significance of circulating tumor DNA in hematological malignancies, a systematic review that was conducted under the mentorship of Dr. Maria Nifatu. Before we begin, I would like to briefly talk about myself. My name is Shaden Alshuaibi. I'm an undergraduate at the University of Birmingham. I'm from Saudi Arabia, a country that has recently been working on the largest construction project, NEOM 2024, and the country that won the right to host the Expo 2030 World Fair. I am a research intern at Cornell University, working in Prof. Emmanuel Zginilis' lab. I'm publishing a paper with Cornell, and you may be wondering, why am I so focused on cancer research when I'm going to be a chemical engineer? Well, the reason to that is because my focus is towards the pharmaceutical side of chemical engineering, and my potential is to understand the concepts of cancer research and apply that in pharmaceutical industries to create drugs and treatments for cancer diseases. And lastly, I have been awarded by the King Abdulaziz and his companions Foundation for Giftedness and Creativity. Now, what are the aims of this research? 1, what are hematological cancers? 2 is to investigate the existing landscape of liquid biopsy in hematological malignancies. 3 is the personalized medicine using this liquid biopsy. 4 is the diagnosis and monitoring of cancer patients using ctDNA. And lastly, and what are the challenges in liquid biopsy using ctDNA? Now, the question is, what are hematological cancers? These are cancers that encompass a wide range of malignancies, originating in the blood-forming tissues, and they play a significant role in oncology. 3 main categories of this cancer are, as you can see on the screen, leukemia, lymphoma, and myeloma. Now, leukemia is characterized by the uncontrolled proliferation of white blood cells, which you may all know as the main blood cancer. The diagnosis of leukemia is by blood tests, mostly the blood count test, which is known as a CBC, and it's mostly observed in pediatric patients. Now, the next thing is lymphoma. It's a cancer that develops either in the lymphatic system, and it's primarily involving the lymph nodes, leading to the abnormal proliferation of lymphocytes. Now, I will move on to the last thing, which is myeloma. And this is the cancerous transformation targets, the plasma cells located in the bone marrow. These specialized cells play a critical role in immune function by creating antibodies. However, when afflicted by the myeloma, what happens is that your immune system doesn't function properly, which causes the individual to be heightened, to be afflicted to infections. How the myeloma is diagnosed is by the urine tests. Now, I will talk about the multi-phased risk factors associated with hematological cancers. They encompass a complex interplay between genetic predisposition and various environmental influences. Notably, factors such as tobacco use, exposure to ionizing radiation, and certain chemical agents have been firmly linked to an elevated risk of developing hematological malignancies. Now, this is interesting because in my religion, Islam, a lot of things have been forbidden. And the reason to why I pursue this passion and read more about hematological cancers is that behind this knowledge, it shows the true meaning of why God forbid things in my religion. Now, moving on into liquid biopsy and personalized medicine. I would like to ask a question for the audience. Does anyone here know what liquid biopsy is? Yes, go ahead. Great, exactly. It's a novel method for analyzing the tumors by isolating and studying various cancer-related components present in body fluids like blood and urine. These components released by tumors include the circulating tumor DNA, circulating tumor cells, exosomes, and tumor-educated platelets. Now, liquid biopsy is a minimally invasive and real-time procedure that may overcome traditional biopsies. Now, you may wonder, what is traditional biopsies? Traditional biopsies carry procedural risk and may not capture the full extent of tumor variability. Obtaining tumor DNA from different sources simultaneously can provide valuable information about gene mutations with potential progenetic or procedure value. The analysis of circulating tumor DNA is the most common application of liquid biopsy in the blood cancers for disease. Now, as you can see in the figure I created here above, the application of liquid biopsies start from taking a blood from a cancer patient and then centrifuging that blood to get the plasma to analyze the tumor DNA. And then we will talk about personalized medicine. So what is personalized medicine? Personalized medicine is the term that commonly used in the media, healthcare systems, and the scientific communities as a whole. This term refers to disease-specific and patient-centered approaches. Now, last thing I would like to mention from this slide is that liquid biopsy's role in monitoring minimal residual disease, which is referred to as MRD, has been extensively discussed elsewhere, but in this systematic review, we will explore the current use of liquid biopsy in hematological malignancies, with exploring the potential future applications, focusing on the personalized treatment for each patient. Now, moving on, I have used all of these literature out there. Those are the databases that have helped me prepare my research project. So the clinical application of liquid biopsy, you either have the early detection or the cancer monitoring during treatment and detection of MRD, which I just mentioned a bit earlier. So for early detection, the approach analyzes the ctDNA, which is referred to as the circulating tumor DNA, and blood samples to identify the mutations of the genes associated with the hematological malignancies, such as the three categories that I mentioned at the beginning: leukemia, lymphoma, and myeloma. The cancer monitoring during the treatment includes a circulating cell-free DNA, circulating tumor DNA, ctDNA collection, and the evaluation of gene mutations. The last thing is identifying the resistant mutations. Now I would like to talk about why is it more challenging, why is liquid biopsy more challenging in hematological cancers than solid tumors? The reasons because of that is that hematological cancers have low ctDNA levels, the background of ctDNA, the disease changes, and the lack of standards and it has a complex analysis. Now, the pre-analytical factors that play an important role in liquid biopsy is the collection. So you have to make sure that you use correct tubes and methods to prevent clotting. And you have to quickly separate plasma to avoid any biomarker loss, which is for the processing. And for the storage, you need to make sure that the temperature is a good temperature. And then for the variability, you consider patients' differences affecting biomarker levels. You of course have to compare and use the right variability. For the volume, you ensure enough sample volume for accurate results. You can't have less, and then that would be a problem. For contamination, you should avoid any contamination, which is like any bacteria or anything touching the surface. And for consistency, standardized procedure for reliable results. Now, for future applications, we have three things I would like to talk about. First of all is the cfDNA fragmentation patterns, which refers to the information regarding fragment length, distribution, and endpoint sequences in the genome after scfDNA sequencing, which carries numerous signals from tumors. And then for the epigenetic features, we have alterations in gene expression that happen without change to the DNA sequence, and then developing multiomics approaches, which makes the test more sensitive, as you can see, by combining from different levels of information. Now, I would just like to briefly say that for the future applications, this was all extracted from the National Library of Medicine, and these are all the references I have used. I would just like to acknowledge Dr. Maria Nifatu, my mentor for this project. I would like to acknowledge Abdulmin Ahmed Rashid and his companions, Humanitarian Foundation, for supporting and funding my research, and my mom and dad who helped me accomplish my goals. If you have any questions, I am open to them, and thank you all very much for listening.
Thank you so much, Shaden. It was really a systematic review. I'll leave the floor open for audience for any questions.
Do you have a question?
If not, I'll have a question.
Okay.
So, how do you manage? I think there goes an extra reading along with your chemical engineering, right? First year of chemical engineering, to do some bits of biology, or maybe a lot of biology. How do you manage it?
To be honest, I would just like to say if you have an interest or a passion, you would be able to manage between a lot of things. Multitasking can be a difficult task, but as long as you go with your inner gut and know what you want to get out of this major, I feel like you can accomplish anything. So time management is all you need.
I do so.
Yeah.
Okay. I wish you all the very best, and thanks again.
Thank you very much. Thank you.